Hello, today we are going to know how the DNA was proved as a genetic material. This is Dr. Hariharan. I am going to tell about how the story of the genetics started and finally how the DNA was proved to be a genetic material. So, through this historic development, there was a contribution of various scientists to prove that the DNA was the genetic material. The story starts from the concept of genetics put forward by Gregor Mendel. So, Gregor Mendel has put forward a theory called the principle of heredity. He has put his findings in his paper entitled Experiments on Plant Hybrids at the Natural Science Society at Brunnos. So he was the first person who brought the concept that the genetic material has been transferred from the parents to offsprings through his famous experiment, pea plant experiment. So a simple example of one of his experiment is that he has taken two characteristics of the pea plants. So one having a dominant characteristics and another having a recessive characteristics. So in this example of law of segregation, he has put up here two characteristics. One is a round gamete, another is a recessive gene, that is denoted by small r. When it is crossed through subsequent generation, that is the first and the second generation, F1 and F2 generation, he has come with certain characteristics. So once he has taken the cross between two parental strings, that is capital R and small r, that is the dominant and recessive. He got a dominant characteristic one in the F1 generation, that is capital R and small r. If it is crossed in between, he got three dominant characteristics and a one recessive characteristics. If the dominant genes goes to the offsprings from the father and mother, then the characteristics will be the dominant. If one dominant factor is more than enough to produce the dominant characteristics, if both the factors are recessive one, then the characteristics will be recessive. This is his findings of his experiment. He has concluded with two basic concepts, that is the various triads, that is the various phenotypic expressions are controlled by a pair of factors. Initially it is called as mental factors, nowadays it is called as genes. This is based upon the genotypic characteristics. One comes from the male parent and another comes from the female parent. So from that the concept of genetics started evolution. So based on this, Walter S. Sutton identified that these paired genes are generally present in its chromosomes of the humans. So in his findings, he has put in a paper that the chromosome is hereditary. So telling about the concept of Mendelian principle of the Mendelian factors that is present in the human chromosomes. So later on, this work was been tremendously put forward, and most of the concept of today's genetics was developed with the help of the Thomas Morgan in 1907. His famous experiments on the Drosophila melanogaster, a fruit fly. He has not only done the characteristics of the putting the genetic concept transferring from parents to offspring, he also come with a new theory called the mutation theory. Based on his mutation theory and the concept hereditary, he can put forward all the basic understandings of the genetic transformation and the mechanism of the hereditary factors. So what he has done is he has taken the fruit fly and he started mutation at certain levels of chromosome. For example, I put with a simple one, that is a mutation in one of the chromosome at BW104. Instead of a normal fruit fly should be in a red eye, it was come with a brown eye. Or a mutation in A99.2, instead of straight wings, he come out with an arc bent wings. So the Morgan experiment has conceptualized the importance of the genetic material, and the characteristics of the offspring is totally depend upon the genetic material present in the parents. So then the studies have started going on. What should be the genetic material, whether it be a protein or it may be a DNA. Because during the time, most of the studies, biochemical mechanism has started elucidation by the chemist. So these concepts started people thinking that the protein might be a genetic material or any other thing. Then the story started moving on, then today we finally come with a conclusion that the DNA is the genetic material. How it has been proved? Let's go through some of the research works put by some scientists. So I will put the first work put by the Frederick Mercer. So Frederick Mercer in 1868, what he has done is he has isolated a compound nuclein from the nucleus of the pus cells. It was containing two chemicals. One is an acidic portion and second is the basic portion. Now we came to know that the acidic portion was the nucleic acid and the basic portion was the protein. So later on, followed by Frederick Mercer's work, one of the great work done by the Frederick Griffith in 1928 on the bacterial transformation. Here what he has done is he proved a theory and confirmed that a concept of genetic transformation occurs between the same species. So he has taken a bacteria. The bacteria generally contains an outer envelope, we call it a capsule. A bacterium which is carrying a capsule is called as a smooth strain, and a bacterium which doesn't contain a capsule, it is considered as a rough strain. So generally the bacterium with a smooth strain are highly virulent in nature compared to the rough strain. So in his experiments what he has done is he has taken a first set of mice. To one set he has given the rough strain. So as you see in the figure, in a rough strain he has injected into the mice. The mice was alive because it is non-pathogenic in nature. In the second group he has injected the smooth strain, that is the bacterium carrying the capsule. What happened is that the mice was get infected and died. So third, what he has done is he has taken the smooth strain and he heat killed it. Then he administered to the mice. So since the bacteria is died, there is no probability of causing infection, so it was alive. Later on what he has done in the fourth set of experiment, he mixed the rough strain and the heat killed the smooth strain. Both concept was identified that the mouse should be live, but the result came out the mouse was dead. So this proves that his concept that the mice causing a certain genetic transformation like that might be happen. The smooth strain bacteria which was heat killed causes the destruction of the genetic material, while once it comes in contact with a rough strain, this genetic material gets transferred. That is called as bacterial transformation and gets integrated with the rough genetic material. Then it expresses a phenotypic characteristic, that is the capsule one. But this experiment was Griffith experiment was satisfactory to a level of that the genetic transformation is happening, that has been accepted and it can be accepted within the species, but there was no proof that the genetic transferring material was the DNA. So later on his work was further carried out by Avery, MacLeod and McCarthy in 1944 to providing a first direct evidence to prove that the active genetic principle was DNA. They have followed the Frederick experiments, that is the same pathogenic bacterium. They have used the Streptococcus pneumonia. What they have done is at the end, after the fourth experiment, the death mice, they have isolated the bacterium. So it was found to be a smooth strain. Later on they isolated the DNA of the bacterium and they have digested with a ribonucleic pancreatic deoxyribonuclease. This is an enzyme which specifically degrades the DNA molecule that we know nowadays. Actually they prove that DNA is the genetic material, not the protein. But this evidence was not satisfactory. So later on, the most concise evidence that DNA was the genetic material was put forward by Hershey and Martha Chase in 1952. They provided a concrete evidence that the DNA is the genetic material. So what they have done in their experiment, that was a very nice one. What they have done is that they have used the tracer technique. So in a tracer technique, they have taken two parts. One is a bacteriophage, a T2 bacteriophage. So a T2 phage is a virus which infects the bacteria. and they have taken a bacteria, that is the host cell, the virus which infects is the Escherichia coli. So the virus has a particular characteristics. It carries only two chemicals. One is an outer caspid that is made up of proteins, and inner is the genetic material, a DNA, RNA. In this case, the T2 virus carries the nucleic acid, that is the DNA as the genetic material. So by using the tracer technique, what they have done is they have made the radioactive phosphorus as a tracer for the nucleic acid and the radioactive sulfur as a tracer for the protein. Then what they have done is they have infected this one into the bacterium. So I will show you in the figure. This is the Hershey's chest. So what they have done is, so this is radioactive coated phosphorus and this is the outer caspid, radioactive coated sulfur. So they have infected this phage into the bacterium, that is the Escherichia coli. Once it has been infected, which one is getting inside the bacterium is the genetic material. If protein gets inside, then the protein might be the genetic material. If nucleic acid getting inside, then DNA is the genetic material. After they found that the radio tracer technique, that is the phosphorus 32 was get inside the cell, not the plasmid, that is the protein which was thrown outside the cell. From this Alfred and Chase experiment, they finally concluded that the DNA is the genetic material because the phosphorus coated was inside the cell. So this is the final history of the Chase and experiment, finally concluding that the DNA is the genetic material, not the protein. So I hope that this historical development, how DNA has been proved to be a genetic material, will be very interesting to you. So I will come with more topics in near future, that how the DNA structure has been elucidated. What are the various scientists, how they put their efforts to prove the DNA structure as the genetic material. So thank you very much for watching the lecture. Thank you.